By way of disclosures, I've recently joined the board of GallMed. I also chair the scientific advisory board at Contravir and consult for a number of other companies, and these are listed here. So Aramchol, or arachidyl amidocholinoic acid, is the centerpiece of GallMed's work, and it's based on 25 years of research and development by the late Professor Tuvia Galat, which led the development of this new class of fatty acid bile acid conjugates, or FABACs. It's a novel synthetic small molecule. It was originally developed for dissolving gallstones. It's orally bioavailable, importantly once daily, and is a tablet formulation. And it exerts its effect through downregulation of SCD1, and I'll discuss this further. Okay, so there were dual mechanisms of action that Aramchol has, targeting two key pathways of NASH pathophysiology. From the steatosis, it reduces the intrahepatic fatty acid content and improves fatty acid oxidation, but it also has a direct effect on liver fibrosis through the reduction of the proliferation of stellate cells and collagen production. And by attacking both of these pathways, there's an impact on liver fibrosis, both for prevention, moving forward in the patient, but also reversal. So what about the downregulation of SCD1? SCD1, or steryl-CoA desaturase, catalyzes the rate-limiting step in the biosynthesis of monosaturated fatty acids, and therefore it's considered a potent step or mechanism in liver steatosis and fibrosis. By downregulating SCD1, you get reduced body adiposity, increased energy expenditure, upregulation of GSH production, enhancing fatty acid oxidation, and maintaining redox homeostasis, and also reduce fibrogenesis in the liver, including AMPK and SIRT. There's been much data demonstrating the beneficial effect of Aramchol on NASH in the literature, and most recently, a review of the role of Aramchol in steatohepatitis and fibrosis in mice, and this was published just recently in 2017 in Hepatology Communications. So what about the preclinical models? Aramchol's effect on the key histologic features has been demonstrated in numerous preclinical models of NASH and fibrosis. The high-fat diet, HFD, induced steatosis and ballooning. The MCD model, the diet which induces aminotransferase elevation and results in a change in hepatic histological features characterized by steatosis, local inflammation, hepatocyte necrosis, and fibrosis. The COL1A1 expression in LX2 human hepatic stellate cells in vitro studies, and then the TAA model in rats. And I'm going to go through the data in each of these models. So in the MCD study design, this was a classic study in which the mice were fed an MCD diet and a control diet, and then they were sacrificed after 4 weeks. The MCD diet included the aminotransferase elevation and changes in the hepatic histologic features, and these changes occurred rapidly. In this particular study, the MCD diet contained 0.1% methionine, and at the end of the second week, after verifying the establishment of NASH, the mice were then treated orally by gavage with Aramchol at 5 milligrams per kilogram per day, or vehicle. And the control diet mice were also treated with vehicle for the same duration, and then at the end of the experiment, the blood and liver samples were obtained. And what you see here is the effect of Aramchol on liver steatosis. Here we're looking at the Sudan staining, and what you can see on the far right-hand side is the staining in the normal diet, and then you see the 0.1 MCD diet where there's significant amount of steatosis. And then when you see the MCD diet plus Aramchol, you see a significant reduction in steatosis, and there was a close to 53% reduction in steatosis with the treatment with Aramchol. If we then look at the effect of Aramchol on the macrophage activation and infiltration, and again, if you look on the far right-hand side in the normal diet, you see the activation in the MCD diet, and then a decrease in activation, both for the F480, the mouse macrophage, and that's on the right-hand side, an 86% reduction, and then in the CD64, a 79% reduction. So Aramchol significantly downregulated and normalized infiltration and activation status of macrophages in the liver. In this slide here, we're showing the impact on fibrosis, the normal diet on the right, and then you see the serious red staining in the fibrosis in the middle, and on the left-hand side is the MCD plus Aramchol, and there was a 43.5% reduction in fibrosis in the liver with the Aramchol treatment. They also looked at the direct collagen production in the liver extract, and what you see in the treated animals was a 38% reduction in collagen, and this is shown here both on the right and the left. If you look at collagen production in the liver extracts, Aramchol significantly upregulated glutathione, and it also elevated the GSH to GSSCG ratio. In the in vitro studies, this is looking at LX2 human hepatic stellate cells, and on the right side is the control, and the left side is Aramchol, and you can see that there was a 41% reduction in collagen production in this in vitro model. And then the TAA model in rats, this is where liver fibrosis is induced by intraperitoneal injections of TAA at a dose of 20 milligrams per 100 gram body weight twice weekly for up to 10 weeks. The rats were treated by gavage with Aramchol at a 1 milligram and a 5 milligram per kilogram per day or vehicle. Control TAA-induced fibrosis were treated with vehicle for the same duration. And then at the end of experiment, the liver samples were obtained, and I will go ahead and show you that data. So on the right, the TAA-treated rats with no treatment, you can see both the microscopic pathology slides and also grossly what the liver looks like. And on the right-hand side with the Aramchol treatment, very different in terms of the amount of fibrosis in the liver, and also the liver looks normal in these rats. And what we saw was a 40% reduction in fibrosis with treatment. So Aramchol reduces key steps in the pathophysiology of NASH, both with the metabolic aspect of the production of steatosis, with inflammation, and with the production of fibrosis. And all three of these aspects are critically important in the development of NASH, leading towards cirrhosis, decompensation, and hepatocellular carcinoma. So what I showed you first was the preclinical development, and now I'd like to tell you where GalMed is in the clinical development. The phase 2A study was completed. This was a multi-center study, double-blinded, randomized, placebo-controlled study conducted in Israel at 12 different sites. The participants all had NAFLD or NASH. They were treated with 2 different doses of Aramchol as compared to placebo, and treated once daily for 12 weeks, and then there was a 4-week follow-up afterwards. There were 60 patients. The primary endpoint was the reduction in liver fat concentration as measured by MRS, and secondary endpoints included the usual liver and metabolic biomarkers. This shows you the phase 2A safety and efficacy data. On the right-hand side, you can see the relative change in the liver triglyceride levels at baseline at the end of treatment. And there was a significant difference on the Aramchol 300 milligrams as compared to placebo in reducing liver fat content. And when one looks at the reported safety events, there was a similar frequency of adverse events in all of the treatment groups. The adverse events were mild or moderate, none were serious, and none of the patients discontinued therapy or withdrew from the study on the basis of adverse events. So based on the 2A study, GallMed moved forward with a phase 2B study. The acronym is ARREST, which stands for Aramchol for Resolution of Steatohepatitis. This is a multi-center, randomized, double-blinded, placebo-controlled study. It's looking again at 2 different doses of Aramchol, 400 milligrams and 600 milligrams, as compared to placebo, in a 2-to-1 randomization. The study involves 12 months of treatment with the once-daily therapy, and then 3 months of post-treatment follow-up. 248 subjects enrolled. The primary endpoint is the reduction in liver fat content, again as measured by MRS, and secondary endpoints include the liver biopsy results, a greater than or equal to 2-point NAS improvement, NASH resolution, and impact on metabolic markers. The key inclusion criteria are similar to many of the studies you've seen in the NASH arena: high BMIs between 25 and 40, the type 2 diabetes mellitus or pre-diabetes, histologically proven steatohepatitis on a diagnostic liver biopsy, either at baseline or within 6 months from screening. All of the biopsies are being read by a central pathologist, blinded to study group assignment. And the liver fat concentrations, the MRS, are being read again, blinded to study group assignment in Israel. The primary endpoint for ARREST is the % change from baseline to the end of study in the liver triglyceride ratio relative to water, and this is measured by MRS. And then the key secondary endpoints include all of the pathology-related endpoints and many of the metabolic in the serum. The enrollment is global, and you can see here where patients have been enrolled globally. Baseline characteristics, about 65% women, the mean age about 54 years. The BMI is, and this is the mean BMI, is close to 33. It's primarily Caucasians with a significant number of Hispanic patients and relatively few Asians or African origin patients. And you can see the baseline characteristics for all of the laboratory parameters on the right. The study enrolled patients with advanced disease. On the right, you can see the NIS scores, and over 70% of patients have an NIS score greater or equal to 5. And on the right is, on the left, is the CRN fibrosis stage, and you can see that 60% of patients have a fibrosis stage 2 or 3. The design and the baseline characteristics as compared to all of the other leading phase 2B studies are shown on this slide, and you can see that there's significant similarity across these studies in the field. The next study, ARRIVE, and the acronym stands for Aramchol for the reversal of HIV-associated lipodystrophy and NAFLD, is a study of Aramchol versus placebo. Is being performed at the UCSD Center, and the principal investigator is Rohit Lumba. So it's a single-center, placebo-controlled study. All of the patients participating have HIV-associated NAFLD. Patients are being treated once daily for 12 weeks, 50 patients enrolled, and the primary endpoint is the assessment of hepatic steatosis by MRI-PDFF. And this study will read out in February of 2018, so those results will be available before the end of the first quarter. In summary, Aramchol is an STD1 modulator. It's shown significant effect on the 3 key pathologies of NASH, the development of steatosis, inflammation, and fibrosis in several preclinical models. The effect on fibrosis is both indirect through reduction of steatosis and direct through the impact on stellate cell proliferation and resulting collagen production. The effect on steatosis has been demonstrated in humans in the phase 2A study that showed a significant reduction in liver fat as demonstrated by MRS. The ARRIVE or the HIV study in NAFLD and lipodystrophy will read out within a few months. And the ongoing phase 2B study, which enrolled a population with severe NASH, will read out in the second quarter of 2018. And finally, there's an end of phase 2 meeting planned to be held with the FDA prior to the initiation of phase 3, and this will be held later in Q3 or Q4 of 2018. So again, I want to thank Ray and the other organizers for the meeting, and I apologize on behalf of my GallMed colleagues that they were not able to attend, but a 5-hour flight from San Francisco is a lot easier than 2 days in travel from Tel Aviv. So, you know, it was not a burden to do this on behalf of my colleagues. Thank you.